Hi, my name is Noah Langenfeld, and I'm currently a PhD candidate here at the Crop Physiology Laboratory at Utah State University. And today I'm going to talk about nitrate versus ammonium, the battle of plant-available nitrogen. Nitrogen is super unique because it's the only macronutrient in plants that is taken up as both an anion in the form of nitrate and a cation in the form of ammonium. And this has major implications for plant nutrition that I'm going to talk about today. So if we first look at the two different forms, on the the left-hand side we have nitrate, NO3-. It's an anion. Left-hand side we have ammonium, NH4+, it's a cation. In terms of uptake rate, there's been a lot of studies that have been done that have shown that ammonium is taken up at a much faster rate than nitrate, on the orders of several magnitudes faster. Ammonium is going to take the cake. In terms of toxicity, ammonium, NH4+, is toxic at a lot lower levels than nitrate. So plants can take up a lot more nitrate and it won't be as detrimental to them as ammonia in the same amounts. So nitrate's going to be better on that front. In terms of storage, nitrate can be stored in the plant vacuoles in cells, whereas ammonium cannot be stored. When it's taken up in the root zone, it needs to be immediately assimilated. It can't be stored in any of the cells. So nitrate's better for this. And finally, mobility. Nitrate is mobile in the phloem. It can be taken up through the roots, transported through the phloem up to the shoots, and stored in those vacuoles, as I mentioned earlier. Ammonium, again, needs to be immediately assimilated. It can't be stored at all. So just based on this, it seems like nitrate might be the primary source of nitrogen that we would want to feed plants. So why do we want to focus on ammonia? So the reason is for nitrogen recycling. Here at the lab, we do a lot of work with NASA on closed life support systems. And we want to try and recycle and recapture as much N as possible. So if we look at N2 gas, that's the major component of air. It's about 70% of air. We found that there are certain bacteria that can fix N from the atmosphere and turn it into ammonia, NH4+. We can then feed that ammonia directly to the plants. Similarly, we have this molecule called urea. That's a waste product of a lot of metabolism. And we have an enzyme called urease that can break that urea down also into ammonium. And both of these can then be fed to plants. So it's kind of a balance between the detrimental effects of ammonium compared to nitrate and trying to feed them as much ammonium as possible because it's easier for us to produce in closed life support systems. And a little bit of the uptake theory behind the two. We see that nitrogen is going to increase the rhizosphere pH for nitrate. That's due to charge balance. So it's an anion, which means if it's taken up as an anion, it needs to release anions for charge balance in the term of OH- or hydroxide. That's going to cause the rhizosphere pH to increase. Ammonium is quite the opposite. So it's a cation, which means it's going to release protons, which are positively charged into the rhizosphere, causing a decrease in pH. At least that's the theory. And for other nutrition elements, so nitrate being an anion is going to promote cation uptake. So these are things like potassium, magnesium, calcium. Whereas ammonium is going to promote anion uptake, things like phosphorus and sulfur that are taken up as anions. So these are kind of the principles I was wanting to test in my studies here. So for my experiments, I ended up growing 3 different crops. I had apogee wheat, so that's a monocot, and it's a grass. Final product was going to be the seed, or the wheat seed itself. Microtinous tomato, that's a dicot, a vegetative dicot, a reproductive dicot. We're going to harvest the tomato fruits. And finally, red sales lettuce, or a vegetative dicot. So a couple different types of plants there. And my kind of experimental setup here, and it was in the research greenhouses. These here represent different columns. So in those columns, I had 3 different treatments. The first treatment was just straight nitric acid for pH control. My second treatment was nitric acid plus ammonium sulfate, so 2 different forms of additional nitrogen in the columns. The third treatment was just sulfuric acid, so just getting sulfate and no additional nitrogen from the columns. So in theory, this one should have been N deficient, this one should have had ample N, and this one should have had plenty of N. The way it worked is I had 3 different tubs. If you can imagine, the plants were growing in these tubs here. And I had them hooked up an electrode to a pH controller. Whenever the pH got above a certain set point, this pH controller would trigger a solenoid that would allow acid from the columns to drip down and correct the pH. And that was the same for all three of these, all independently controlled. Here's a couple pictures of my setup. See here, I've got 6 different tubs. So I was growing lettuce and then tomatoes. We've got the pH controllers right here, air supply, and then supply of the acid. In the middle, that's what the tank looks like. So the plants grow on top of the tank, on top of these styrofoam rafts. The roots drape down into the liquid below. We've got my pH electrode here, sensing pH constantly. And then finally, we have the columns. So these are the reservoirs essentially for my different treatments of acid. They have nice little graduations here, so I can easily tell the amount of nitrogen that has been applied through the columns over a certain time period. So if we look at some of the results here, so the tomatoes, they haven't been harvested yet, but they're at a mature enough stage here. So if I were to ask you to pick out the treatments, it would be really hard to pick out the treatments. They all look pretty similar just from a main point of view here. But the actual treatments are, this was the nitric acid treatment, so just straight up nitrate. This was the ammonium sulfate treatment, so nitric acid plus ammonium. And this was the sulfate treatment. So like I said earlier, this one should have been indeficient. But if you look at them, they all look pretty similar. There's not really much of a visual difference that at least I can tell between the three. Lettuce, we had a similar story. So again, look at all three of those. Not really any visual difference. The treatments were exactly the same as before. We had nitrate, ammonium, and then sulfate. So we were kind of surprised that even the sulfate one, which we knew was going to be N deficient, visually looked exactly the same as the other two treatments. Let's look at a couple of the harvest parameters here. So I looked at both lettuce and wheat, because I mentioned the tomatoes are still growing, but lettuce and wheat were grown out to full maturity. For yield, in terms of dry mass here, we have lettuce, which decreased slightly as we increased the percentage of ammonium that was being applied, whereas wheat increased slightly. These 2 points here represent the low N, or the sulfuric acid treatment, so no additional N from the column, and as expected, their yields are lower for both species. For percent root mass, we have, look at lettuce here, almost identical across the board as we went from no additional ammonia to 32% applied as ammonium. Wheat, we had an increase in percent root mass. So that means that the plant was putting out more roots, trying to somehow get more nutrients. Look at the different the sulfate treatments here. We had opposite effects, where for lettuce it dropped below, for the wheat it went above. So kind of different things going on with the species there. Harvest index, the lines are flat, but I think that's one of the most interesting things. For both lettuce and for wheat, the harvest index was nearly identical across all three treatments. And for the lettuce, again, that was the mass of the shoot or the leaves that you would eat. For wheat, that was the mass of the seeds that would be used for the grain. Look a little bit at EC and pH here. So I measured EC, or electrical conductivity, so the salt content of the solutions towards the end of the treatments. Just for comparison, tap water, Logan City tap water, has an EC of about 0.4, whereas the nutrient solution that we're feeding them has an EC of about 0.9. So if you look at the wheat here, we see it's above 0.9, 1.2, 1.3, which means that the wheat was accumulating salts in solution at the end of its life cycle. Whereas lettuce is about 0.6, so it was going through those salts, I'm using them up, still not terribly low. But then we look at the tomato here, especially this one in particular, 0.24. Wow, that is way below that 0.9. So if you were to show that number to any other plant scientist, they would think those tomatoes were really suffering. But as we saw in those pictures, they look exactly the same as all the other ones. And the other two treatments were also the lowest out of all three species that I found. And finally, pH here. If we just draw a couple of quick graphs, we have pH here on the y-axis and then time on the x-axis. For wheat, the pH was pretty stable across the entire growth cycle. Of course, they have pH control, but that's only pH control down. There's no pH control up. Lettuce, same thing. Didn't really have any issues with pH control at all. Tomatoes is where it got a little weird. Initially, we had no problems. But then, so we had some of the tanks start to drop in pH, which would be expected, especially for the NH4 plus tank. So it was releasing protons into the solution, dropping the pH, which is what we saw. So then I would have to add some base, KOH, to bring it back up. Then it would drop again, add some more KOH, and so on until it leveled back out. And then just within the last couple weeks, all three of the tanks started doing that. About 2 weeks ago, I had that issue. So it wasn't just the ammonium tank this time. We had a couple other tanks going wonky. Not quite sure why that's happening. They are in a heavy reproductive phase right now, so that might have a little bit to do with it. But definitely interesting results for the tomatoes and pH. And finally, we're going to talk about the tissue analysis. So the USU analytical labs had them analyze the tissue for the tomatoes and the lettuce here. Lettuce I broke down into young and old leaves, just because if we look at some of the differences here, particularly things like calcium and potassium, there's major differences between young and old leaves in those elements. Potassium, for example, we have about 3 to 4% potassium in young leaves, whereas old leaves, wow, over 8% potassium in those leaves. So big differences. Similar for calcium, much higher calcium content in the old leaves than in the young leaves. But all that aside, if we actually look at the different treatments, we're expecting my nitrate treatment, so that's an anion, it should be inhibiting other anion uptake. For these macroelements, that's phosphorus and sulfur. So I would expect phosphorus and sulfur to be the lowest out of those three, which it was for the young, and also was for the old leaves. So lettuce kind of followed that trend. If we look at tomatoes, again, should be the similar thing, but it was the lowest here, which, yeah, but here it was kind of the highest, kind of opposite what we were seeing for the phosphorus in tomatoes. So there definitely seems to be some species-specific differences for all the parameters that we're looking at here. If we look at the ammonium treatment, so ammonium being the cation, should inhibit other cations and promote the uptake of anions. If we look at the group of cations here, potassium, calcium, magnesium, these should be the lowest. At least that's what theory would say, but that wasn't necessarily the case. Magnesium, there was no difference. Calcium, it was actually the highest, and potassium, it was right in the middle. So conflicting results there. For the young leaves, old leaves, this was actually the highest. These were all the same, and again, this one was the highest. So kind of almost opposite of what we would expect. Tomato here, we have a similar story. So cation should be inhibiting the cations, but it was actually higher in this case than my nitrate treatment. These were all about the same, and this one was actually the lowest. So there's differences between the actual element themselves, whether they're divalent or monovalent, and then between the species as well. So to wrap things up, just a little bit of a conclusion of what I found, theory versus results. For nitrate, we expected to increase the pH of the solution. I definitely found that for the lettuce and for the tomatoes. Just anecdotally, if you walk out to the greenhouses, you'll be able to stand by that nitrate-only column and you'll hear the solenoid click on and off every couple minutes, which means that it was calling for acid because it was increasing the pH. So that I would say was definitely true. Promoting cations, not so much. Like I just talked about, it didn't seem to really promote the uptake of cations in either lettuce or the tomatoes. Whereas inhibiting anions, it did kind of inhibit some of the anions, again, depending on the species and the actual anion itself. For nitrate here, or sorry, for ammonium here, decreasing the pH, we did see that, especially in the tomatoes, like I talked about, where it was dropping the pH down. I had to add some KOH, some base to bring it back up. Didn't really see that in the lettuce or the wheat. Not quite sure why that is, and could be because the lettuce didn't put enough load on the system. Lettuce was only in there for about 30 days. The tomatoes have been in there for over 100 days now. So it could be differences in their life cycle. And similar to the nitrate here, for the cations, we didn't see any inhibition of cations. So nitrate didn't promote cations, ammonium didn't inhibit cations. Not quite sure what was going on there. But the anions, it did end up on promoting and inhibiting. So with that, I'd like to thank the NASA CUBES program for funding this research, of course the Crop Physiology Lab for providing the facilities and equipment, Dr. Bruce Bugby, my advisor, for helping me design the experiment, set everything up, and then Alec, Paul, and Logan for helping me troubleshoot and get all set up with the experiment. With that, I'd like to say thanks for watching.